Mr. Subhan to have his opening speech now. Please.
Okay, thank you, Dr. Firsan. Good afternoon, everybody. Firstly, I would like to thank God who has given us blessing and mercy so we can attend to this online seminar program in a good conditions. On behalf of Faculty of Pharmacy, Hasanuddin University, I would like to thank to our speaker today, Dr. Dipti Trivedi, National Center for Biological Science, Bangalore, India. Thank you for your time and we'll share about engineering Drosophila genome. We are really appreciated for your valuable time, and I hope we can make research collaboration in the near future. And also I would like to thank to Mr. Firsan Nainu and team for arranging of this seminar. I hope we can learn more about this topic, and of course it will give more impact to all of the participants on this seminar. That's all. Thank you very much and have a nice seminar.
Thank you.
Thank you, Dr. Firsan.
Yeah, thank you very much, Dr. Subhan, for the nice opening speech. So I'm sorry, there's a little technical problem in my laptop. Okay. Great. So we will have an introduction session now. I will introduce Dr. Trivedi to everyone here. Please let me have.
It's okay to turn off my video.
Yeah. Yes.
Okay. Thank you.
Thank you. So Dr. Trivedi actually received her PhD degree from University of Cambridge in UK, where her research was focused on Drosophila genetics, which is genetic basis of phototransduction. She received the prestigious Gates Cambridge Scholarship for her doctoral studies. Following this, she did her postdoctoral studies in University of California, USA, in mouse and Drosophila genetics, studying intracellular trafficking in rod cells and motor neurons, respectively. She has extensively used Drosophila and mouse genetics, including genetic engineering and transgenesis as tools to understand fundamental biological phenomena. Dr. Trivedi joined CCAMP NCBS in December 2013 as a technology scientist to develop and establish new technologies in Drosophila genome engineering. So I think her credential is really great. So we will learn so much from Dr. Trivedi for today about genetic engineering using Drosophila. Great. So I can now assure that I can let Dr. Trivedi take the session from now. Please, Dr. Trivedi.
Thank you, Dr. Subhan and Firsan, for the great introduction and starting. I will, okay, I'll just close the video so that I can see my screen. Yeah, okay. But you can see me, right?
Yes, we can see you.
Okay, great.
Yes,
so, okay. Today I am going to talk about my favorite model organism, Drosophila melanogaster, and how people use genome engineering to address some important questions in cellular and molecular biology in flies. I will be using the term Drosophila and flies interchangeably. All the time I mean only one species of Drosophila, which is Drosophila melanogaster. So bear with me on that because we tend to use this term very loosely. So I work at National Center for Biological Sciences, Bangalore, India, and I head the fly facility there, where we make transgenic flies as well as genome engineered flies as a service to different international and national users. So I would first, because this is, I have been told that this is an audience which is majorly constituted of undergrad students. I would urge you to ask as many questions as you would like, and I would try to start with introducing the system that I'm working on, and then also introduce CRISPR-Cas9. Following that, I would I would talk about how genome engineering is being used in Drosophila melanogaster, while giving specific examples from our own research in the meanwhile. So I don't know. Okay, yeah. So as we all know, we are all different species, humans as well as other species, we are all linked through temporal evolutionary tree, and the closer we are on the evolutionary tree, the more the similarities we have. So if you look at this evolutionary tree, we have the closest. So for example, all the plants are here, all the mammals are here, then yeast is here. So we are all linked and we have very similar organizations, and the closer we are, the more similar we are to each other. So here is us, that is humans, and here is Drosophila. And although we are not as close as we are to mice and guinea pigs, for example, but there are very many similarities between the flies and human beings. And this is shown very nicely by Professor Andreas Prokop in this slide, where we see that we have very similar body patterns, for example. So we have here that the way our head, body, and thorax, abdomen is formed is very similar between flies. So we also have head on the top, then the thorax, then abdomen, and so on. And similarly in flies and humans. Also, we have similar left-right symmetry as flies. So our left side is very symmetrical to the right side. So that's one part of development, but also our flies have a very complex nervous system with distinct regions of the brain doing distinct functions. So flies are able to learn, to remember, to see, to taste, to smell, using distinct brain regions, and are able to coordinate these different senses and different functions together. Also, flies have a digestive system, which is very similar, circulatory system, excretory system, muscles, and so on. However, if you look at the genome of flies as compared to humans, and this is a kind of old slide, but it brings in the point that fly genome is much smaller as compared to human genome. And here you see it's 23 times smaller if you look at sequence by sequence. But when you look at number of predicted genes, it is only slightly less than half the size. So the number of genes are quite substantial, actually, which suggests that the genome is very compact, and the intergenic spaces as well as introns are much smaller in flies as compared to humans. Also, there is much less, probably there is much less redundancy in the genes, which means that for one gene, for one function, there are not many genes for the same function, but usually one gene that codes for a protein for one function, unlike humans, where there is much more redundancy. That makes flies a very good model system for genetic studies. In addition to its obvious advantages of shorter lifespan and is there a question? Okay. So in addition to its obvious advantages, which is shorter lifespan and its size and ease to grow and and cheap methods of growing it. Because of these reasons, flies have been used as a genetic model organism. Now, to use any system as a genetic model organism, what one needs to do is to mutate the genes that you mutate their genes, and then to figure out what defect this fly would have after this mutation. So over past 100 years, Drosophila biologists have done just that. The early mutants that were being used by Thomas Morgan, who was actually the one who introduced flies as a model organism, were mostly spontaneous mutations, meaning that he and his colleagues were just trying to find within their bottles of millions of flies if there was one fly that had some kind of difference. And these were completely spontaneous mutations. But flies were the first one to actually being mutagenized by X-rays and chemicals, and Professor Hermann Muller got Nobel Prize for this, where one could actually mutagenize the flies at will and then figure out roles of different genes, and this has been used till now in the forward genetic screens, when you randomly mutagenize flies and then try to find out specific phenotypes and how and what are the genes that are involved in these specific phenotypes. However, as you can see, if you do a random mutagenesis, it's very difficult, especially if these are point mutations, very difficult to find which gene is responsible for this. A p-element based mutagenesis came in 70s, where one could actually insert these p elements, which are larger DNA chunks, into the genome and hop them around in the genome, and then try to figure out if there are any defects. Now, this was again a random mutagenesis method, but at least because it was a bigger piece of DNA, one was able to try to chromosomally localize it, or even localize it in what part of the chromosome this p element existed, and that made this method slightly easier. However, it was still not done at will on a particular gene. And the final one in this category, random mutation, is the transgenics, which could be basically inserted into the genome. Although again, these insertions were mostly random in the beginning, while we are able to do them now in a very specific way. But all these methods were very useful because one could generate these enhancer trap lines, as well as mutants, protein traps as well. Then came the homologous recombination or gene targeting. This was the very first time it was possible to actually create point mutations in the gene exactly where you want them to be. This was a very cumbersome and labor-intensive method because you will have to then make these huge homology arms, usually 3 to 5 kb, and the efficiency was much low. So although this was invented in 90s, there is not much use of these throughout now as compared to what the newer techniques are able to do. They were, for example, never made a genome-wide things using these homologous recombination, and were done on gene-to-gene basis. That was RNAi actually was the first thing that revolutionized the way one could address specific gene problem, but at a larger scale, because these transgenic lines which express RNAi actually the first. Yes, is somebody asking question? Okay. Okay, so RNAi was the first time when one could do it in a genome-wide manner, and we have large number of RNAi transgenic lines available, which can be used to knock down. So these are not knockouts because it's not at genomic level, but at RNA level. But one could knock down genes, very specific genes, by this method. Although there were problems with off targets and so on, but let's not go into that part. So truly at the very specific genomic locus, the techniques that could be done in a much more easier and more efficient way came only when zinc finger nucleases, TALENs, and CRISPRs came, and this is a very recent invention after especially CRISPR, because it makes things much more easier as compared to the ZFNs and TALENs, which require a lot of pre-injection stage where you are actually making the constructs. They are much more difficult in case of ZFNs and TALENs. It's much easier in case of CRISPR. So unlike other systems where CRISPR is being used, especially the non-model organisms and higher organisms where resources were not available, flies actually came with a huge repertoire of tools, genetic tools from past 100 years, which are being used by the Drosophila biologists. We have been very lucky to have these stock centers where all these tools are available, and any Drosophila biologist can ask them and use them. So we have large collection of mutants, of chemicals and transposons, and we can do tissue specific analysis of mutants and tissue specifically express things, and study a lot of things by very cool genetic techniques. However, CRISPR has basically added a feather to the hat of Drosophila genetics. So what we are able to do even before CRISPR came into picture is that we can express genes, specific genes, even if it is coming from humans, for example, or any other organism, in a tissue specific manner, in either females or males specifically, only in embryos or different larval stages or pupae and so on. So we can basically very specifically restrict the expression of genes. We can also reduce the level of transcripts at a very specific time point. So these are the things which we are able to do very specifically in flies, even from before, because of the techniques that are already available. Now I will try to introduce CRISPR-Cas9 technology and how it has actually enhanced what is already available in flies and how we can generate mutants with them. But before I go into that, I thought I'll just very briefly explain what exactly is CRISPR-Cas9 technology and what is the history of it and how it came to being. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. As the name suggests, what it means is that these are very short palindromic repeats, which are actually shown here as these triangles, facing triangles, showing that these are palindromic and they are repeated. So you see that they are again and again repeated. And these are clustered. So these clusters are called CRISPR arrays, and they are interspaced, meaning they are not together, but there are spaces in between. And these spacers, I'll get back to what these spacers are, but these spacers do not belong here, but they are achieved or acquired. And in addition to this CRISPR array, are other CRISPR-associated genes which are present. Now, in 1980s, it was found that in bacterial and archaeal genomes, so prokaryotic genomes, these arrays were present there. By sequencing, they had found them, but they did not know what exactly they are for. So these were only sequenced and left like that in 80s. It was only in early 2000s that it was found that these are actually important for acquired immunity in flies. Yeah. So how does that work? So bacteria, just like us, is not always healthy. We also fall sick, and so does bacteria. And the way bacteria may fall sick is by some phage infection, or if, for example, transfer of plasmid DNA from other bacteria. So it does recognize the foreign DNA. And what it does is that it has a system, it has a complex of proteins that recognize these DNAs as foreign sequences and process them, cleave them into these small DNA pieces, which are called spacers. And these spacers are then inserted into these CRISPR arrays between these palindromic repeats. Okay, so these, which means that every time there is a viral infection or some foreign DNA entering the bacteria, one thing is, if there is an infection, the bacteria may die, and that's the end of the bacterium. But when the bacteria is not dying, it is actually cleaving this viral DNA and remembering in its genome that this particular piece of DNA, it has seen it before. So just like how we get immunized in our system. Now, what happens that this is kind of acquired immunity, because when the viral infection happens again, this CRISPR array is transcribed into something called pre-CRISPR RNA, which is then processed. So it is chopped off and processed into something called CRISPR RNA. And the other proteins which are also present in this CRISPR locus, gene for which is present in this CRISPR locus, are then together with this CRISPR RNA is able to recognize by its exact sequence by which this spacer was present. And once this exact sequence is found, a protein called Cas protein is able to chop that foreign DNA into pieces. And that's the immunity phase. Now, you may ask that why this CRISPR RNA, these spacers are also present in bacterial genome, and they are also present in the viral genome. So why is it not able to cut itself using the same machinery, like how we have autoimmune disorders, right? So that does not happen, because when in the beginning it is cleaving this novels and making these novel spacers, it cleaves this plasmid DNA or viral DNA only where there is a specific sequence which is called a PAM sequence, protospacer adjacent motif, and it acquires only this piece of DNA in its genome. And when it is trying to recognize the viral genome, it's actually looking for this PAM sequence, which was originally adjacent to this spacer sequence. So that's how it only recognizes the viral genome and not its own genome. Now, once this thing was found, which was in early 2000s, very quickly, in vitro experimentalists figured out that if this particular protein called Cas protein, which is Cas9 in this case, there are many Cas proteins available, when this Cas9 is present with the CRISPR RNA and another RNA which is also coming from the same locus, which is transactivator of CRISPR RNA, if these three are present, depending on what this sequence is, it is able to recognize any DNA sequence, provided it has the PAM sequence next to it, and is able to cleave this DNA sequence in a double-stranded manner, leading to the cleavage of target DNA. It has, this Cas protein has two functions, one that cleaves the target sequence and one that cleaves the complementary to to the target sequence. So all what you need is a Cas9 protein, a CRISPR RNA, and a tracer RNA. And the target sequence having the PAM sequence, which in most of the cases is NGG, NGA, NAG, something like that, and it is able to cleave it. Now this is very useful. This was done in vitro experiment. This is very useful because now if you do the same experiment in a cell in vivo, what would happen is that once this cleavage happens, the cell's machinery of either non-homologous enjoining or for homology-directed repair, it will immediately kick in. And because this non-homologous enjoining is not precise, it may lead to small gene disruptions by either small insertions or deletions and lead to cause of mutation in the specific locus against which you had targeted your guide RNA. Now, guide RNA, which I have not mentioned just now, is actually a combination of CRISPR RNA. It's a fusion of CRISPR RNA and mature tracer RNA, so that you don't need to have two different RNAs. You can actually use a guide RNA, which is fusion of these two. So all what you need is a guide RNA and Cas9 and the guide RNA specific for a sequence, and you are able to cleave it, leading to non-homologous enjoining or homology-directed repair. Now, this homology-directed repair, which I had talked to you about, this homologous recombination, that because there was no cleavage to begin with, the cell is not very open for this homology-directed repair, leading to very low efficiencies. But once the genomic DNA is cut, now your homologous recombination machinery is very active to be able to insert the DNA in homology-directed manner. So this is quite useful and has increased the efficiency of homologous recombination, which was being done in flies before or other organisms before, but this has made it much more efficient. So what can one achieve? We can have genomic deletions, we can make SNPs, small nucleotide changes we can make. We can do knock-ins by homology-directed repair. We can replace genes, especially useful when you want to see whether a human gene, it has the same function as the fly gene, for example. And you can do genomic rearrangements, and you can also do tissue-specific work, which I will talk in details in a bit. Also, one can do multiplexing, which means that you can have several guide RNAs and then mutate several genes at a time. I will also get into that. But now we'll go to Drosophila and how one can make the use of CRISPR-Cas9 in Drosophila efficient. Now, one thing I want to mention here is that Cas9 protein is coming from prokaryotes, bacteria. Many of these bacteria, when we grow them in labs, they grow well at 37 degrees. And Cas9 in vitro experiments also suggest that Cas9 functions the best at 37 to 39 degrees. While Drosophila, as we know, we don't grow them at such high temperatures. In fact, flies are grown at 25 degrees, and at 29 to 30 degrees, they are already not very happy. So we certainly need to make this system more efficient. So that there are three parameters that we can consider. One is the Cas9 protein itself, then the guide RNA, and the method of delivery. So we'll go to Cas9 and guide RNA first. So let's go to Cas9. As I said, Cas9 comes from prokaryotic systems, which means that because prokaryotes have a very different codon usage as compared to eukaryotes. So the first step which was done to make this Cas9 efficient is codon optimization. So Cas9 codons have been optimized both for humans as well as Drosophila, and both these codon optimized proteins or genes have been used in flies with equal efficiency. equal efficiency, but it has also been seen that when you codon optimize Cas9 for Drosophila, especially when you are doing tissue specific things, the expression is too high and it can have its own phenotype. So most of the time, people in the fly people use human codon optimized Cas9. Then also what concentration of Cas9 to use, and that has also been worked out, as well as promoter usage. For example, if you want to express the Cas9 in the entire body, you can use promoters like actin promoter or tubulin promoter. However, if you want to mutate a very essential gene, which you don't want to use a promoter that's ubiquitous, because then the progeny will not come out, because if the gene is essential and if it is mutated in the whole body, the fly is not going to survive. In that case, mostly for making a germline mutation, germ-specific promoters like nanos or VASA are used. In addition, we also sometimes use UAS promoter, for which I'll come in a while. Now, when we talk about guide RNAs, we are of course looking for the concentration of guide RNA. It has been shown that changing the concentration and making the guide RNA concentration higher increases the efficiency. That's not true actually for Cas9, that beyond certain concentration of Cas9, it doesn't make a huge difference. But increasing guide RNA concentration increases the efficiency, but it also increases lethality. That's just because of the too much of DNA, I guess. Promoter usage, because it's an RNA, we usually use U6 promoters. There are 3 U6 promoters, and it has been worked out which promoter works at the highest level and which promoter works at the lowest level. But sometimes having highest is not the most important thing. You may have tweak what promoter will work the best in your system. The third and most important part of this is gRNA specific sequence, because these sequences are very important actually. And I will get to this in a couple of slides, that how empirically and theoretically various softwares have come up, where we know now that which are the sequences which may work better as compared to the other. And now the method of delivery. So in flies, usually most of the delivery is done at a very early embryonic stage where cellularization has not happened, so that DNA can directly access the nuclei and target the gene. So the first method here is Cas9 mRNA and guide RNA. So both are in RNA stage, and you inject it in wild type flies and then look for heritable mutants in the next generation. Now this is not widely used, although it's highly efficient, is because RNA are not very easy to handle by most of the labs, and it usually degrades and so on. So it's not a very robust system as such. Second one is Cas9 plasmid and guide RNA plasmid. So both are in plasmid form and they are injected in wild type flies. This is very useful because this is both in plasmid form is very robust and but at the same time, because both of them are plasmids, that means that both should actually enter the genome, and in every cell both should be present. So efficiency in this case is kind of lower than what I will discuss in the later points. However, it's very useful in the situations where this wild type fly that you're dealing with is a specific genotype that you're interested in, and you don't want to make a lot of genetic combinations before you start your experiment. So it's a useful system and many people use it, and we also use it. The third is, which is something which is used the most often, is that you have a transgenic fly that already expresses Cas9, and there are several such transgenic flies which are available in stock centers, and you can just grow them and inject a guide RNA plasmid. So this is the most quickest way of doing it. It is much higher efficiency because Cas9 is present in all the flies, all the cells, and all the nuclei, and you just inject one thing, so you make sure that that goes into the nucleus. Now with this one, the disadvantage also is that once you inject the Cas9 plasmid, there is a lag at which Cas9 would work. So Cas9 plasmid will make Cas9 RNA, then Cas9 RNA will make Cas9 protein, and that's the time when the mutation will start to happen. So you will have in this first generation a lot of mosaics. Although second generation, you will get heritable mutations. Here, because you just have to convert this sgRNA plasmid, transcribe it to RNA, you will have many more cells in this generation that have got the mutation because Cas9 is being expressed from the very beginning. The fourth one, which is much more efficient than any of these three, is when you have a Cas9 transgene as well as guide RNA transgene. It's easier for any lab to use which do not have any of these injection methods to use. So what you have to do is you just take these two transgenic flies and cross them and then look for heritable mutants. So this is, although in the beginning to make these transgenics, it can be an effort, but once these transgenics are ready, you just have to cross. At this point, I would also like to say that there are several labs, actually 3 or 4 labs, which are very generously making these genome-wide guide RNA transgenic flies and making them available for any Drosophila biologist to use. So all you have to do is get these guide RNA flies, Cas9 flies, and cross them for your gene of interest. As I said, I would now talk about how to choose an efficient guide RNA target. There are many experiments that have been done, which I will not go into, but there are algorithms now based on a lot of empirical data of thousands of guide RNA and their efficiencies, and then looking at their structures and what kind of secondary structures these RNAs will make, and so on. And this is just one example of one of the study where they have shown that having GC-rich region right next to the PAM and at the tail end of the sequence is much higher efficient than having TT stretches, for example. So this is just one example, but people have done these empirically and come up with many of these algorithms which are freely available, and anybody can go to these websites and use them. So things you should remember while designing your own guide RNA is that you should look at the actual organism in which you are going to make these mutations and sequence it, because the flies that I have, for example, are going to be very different from flies somebody else in Indonesia has, or somebody else in Germany has, or and so on. So it is because they have been reared in our labs for generations, and they would have acquired their own mutations and so on. So it's best to sequence the region of interest and compare it with the genomic data that is available already, and then design the guide RNA. Also, not all guide RNA are active. Not all guide RNA that are active in one system or one tissue are active in other tissue. That is because the way the genome is wrapped around in different tissues. and all cells do not behave similarly for the same guide RNA. This is just one example which we have done where this is actin Cas9 crossed to guide RNA against white gene. And white gene is actually expressed in Drosophila eyes, which give red eye color to the flies. Now, because actin is expressed everywhere in the genome, essentially you should have seen using this guide RNA completely white eyes, but that does not happen. And you can see that there are a lot of red patches here, which means that the CRISPR has either not worked in these cells, or it has worked, but it has mutated very small region which is in frame. So there can be some in frame deletions which keep the function of the gene itself. Multiplexing has also been tried in flies where 4 genes at the same time have been mutated. But as you can see that the efficiency of single mutations is much higher as compared to if you are mutating 4 genes at a time. And this particular experiment is done with very visible phenotypes. So if you were doing something which was not visible and you have to sequence every time to figure out whether the mutation has happened or not, making these kind of mutations may be not that easy. Now I'll go to tissue-specific genomic manipulations, for which we have a system already ready and highly widely used, which is the UAS GAL4 system, where we have large number of these enhancer trap GAL4 lines which express in very specific tissues, and they are available, hundreds of these lines are available, which we can use to actually express any gene of our interest, provided we place it under an upstream activating sequence. Upstream activating sequence will not allow the gene to express at all unless a GAL4 driver is expressed as well. So, so this gene will then express, then become conditional and will be expressed only when in the tissues where this genomic enhancer functions. So by this, what we can achieve is that if we have a GFP, let's say UAS GFP, and we use a GAL4 that expresses only in eye, then you see that the GFP is very specifically expressed in the eyes. So this system has been used, and UAS Cas9 flies have been used to mutate the genome in a very tissue-specific manner. And these are some of our data where we have used tissue-specific. These are some tissue-specific knockout examples where we have used eyeless GAL4 that expresses only in the eyes, and several genes here. And many of these genes actually, when removed in a pan-organismal level, are lethal. So you will not get flies at all. They will probably die at embryonic or larval stages. But here, what you see is that when you mutate these genes specifically in eyes, you see various kinds of phenotypes. And these kind of screens then can be done to see the role of these genes in specific scenario. Here we have just compared RNAi with CRISPR-Cas9. You see that PI3 kinase RNAi does reduce the size of the eye, but when you compare it with what a guide RNA CRISPR-Cas9 can do, you see that the eyes become much smaller, which is consistent with the fact that here you are removing the entire PI3 kinase at the genomic level, while here you are just removing the transcript, and it's probably not a complete removal as well. So there are 2 different guide RNAi lines here. And this is another example. So what we can achieve by tissue-specific things is very good, but there are some issues, which is that not all cells may get the deletion, as I had shown earlier. And we don't know which cells got the deletion and which did not get deletion. Now, solutions can be using more guide RNAs or marking the cells that got the deletion and so on. Recent work by Philip Port et al. Has shown that some of these problems actually can be taken care of. So I'll just go through this slide a bit. So when look at this cell population where the gene is completely expressed, and this is the genotype plus over plus. And what we want to achieve is that we want to remove the gene specifically in certain cells while keeping them in other cells. That's exactly what we want to achieve. And by using ubiquitous guide RNA, which we have been using so far with a conditional Cas9, this is what we get. So it's not a very clean system as such, but if we use conditional guide RNA as well as conditional Cas9, what they were able to achieve is much tighter regulation of guide RNA and much better mutation rate. So this is something which can be kept in mind and can be used in the future. The second part, so this whole thing was about non-homologous end joining. The second part is homologous recombination. By homologous recombination, one can achieve knock-ins, tagging, enhancer and protein trapping, and specific point mutation. So these all things are possible if you also inject along with your guide RNA and Cas9 another piece of DNA which can homologously recombine with the genomic region. Now, what kind of DNA would you insert? There are two ways of doing it at this moment, and that is SSODN as well as plasmid based, and I'll go to them one by one. So what is SSODN? These are single short oligodeoxynucleotides. These are just exactly the oligos that you use for your PCRs, but slightly longer than that. They are usually 100 to 200, up to 400 to 500 bases long. They can be easily synthesized by a company, so you do not have to do anything, you just order them. They are useful for small base pair level changes. You can have small insertions like 50 base insertions or so on. But the issue with this is that because there is no visible marker as such, your screening is usually by PCR and sequencing. So these are the steps that we use, and this is one of our own examples, where you can just design the guide RNA, design the SSODN, inject both of them in a Cas9 fly, and do single line process. So you will have to do single line process, and then you PCR and sequence, and you see that this particular GAT has been now converted to a AAA, and this is exactly what we wanted using this small oligonucleotide. So this can be very cumbersome at the screening stage, while it's easier at this stage, early stage. Plasmid based is actually just the opposite. It's more cumbersome to make it, but screening can be much easier. So what you have to do is you have to clone, and I'll give you one example. For example, this one, which is coming from Graz et al., where you have a plasmid which has two homology regions. So one homology region here and the other here, where the guide RNA cuts in one place, and then you can actually replace this region with whatever you want to replace it with. Usually it comes with a marker. So in this case, it's 3xp3ds red, which expresses red in the eyes. So once you have done the homologous recombination, all you have to do is go to a fluorescence microscope and look for red eyes and fluorescent red eyes. And the fly which has fluorescent red eyes is going to have the mutation. In this case, they are adding GFP, but you can do whatever you want to the genome in that region. And once you have found that your fly, you can actually use piggyback transposase to remove this 3xp3ds red, leading to only the mutation or addition or insertion that you wanted to do. And this, in the screening stage, is much easier.
Hello.
Koneksi putus Pak Firsan dari Fedi.
Oh, oke. Saya pikir saya tadi. Tunggu, saya hubungi lagi Dipti ya.
Yes, betul-betul.
Oh iya. Mohon maaf Bapak Ibu, mungkin koneksinya terputus dari Dipti. Nanti akan segera disambungkan lagi. Halo Dipti. It's off. Your audio is off, Dipti. Your microphone.
Oh,
yeah.
Sorry.
Sorry.
Yeah, I'll start again. The electricity went and came. So.
Yeah. Thank you.
Okay, so. Can you see this now?
Yes, I can see. Maybe everyone can see as well.
Okay.
Yeah.
Okay. So you can basically mutate whatever you want and remove the 3ds red at some point later. However, as you can see that the initial part in this particular case is time consuming and it takes a lot of effort and a lot of troubleshooting is required when you are cloning it. Very, very recently by Perimon Lab, they have introduced this new system, which has been tried actually in mammalian system before, is something called CRISPR-Cas9, and that's homology-independent insertions, which is quite interesting because you don't need actually any homologous recombination. You basically have a donor plasmid, which has a marker and anything. So here they have given an example of T2A gal4, but you can actually add anything you want here. And it also has something called a CRISPR-Cas9 target site. And this donor plasmid can be inserted by injection into a NOS-Cas9 embryo. Now what, along with a guide RNA, which is against this CRISPR-Cas9 target site,
Ya, sepertinya koneksi dari India mungkin ya.
Iya, bermasalah Pak Firsan.
Iya. Dipti masih masuk nggak ya?
Which are linear and hello.
Hello, we lost you a little while. Sorry.
Oh, okay. I think there is some problem here.
Dipti, can I suggest something? Maybe you can turn off. Maybe you can turn off the video to reduce the. I'm not sure if that helps. Yeah.
Yeah.
But we can try.
I need to see how to turn it off. Stop video. Okay.
Yeah. Okay, great. Thank you.
Yeah. Yeah. Okay. I'm actually going to finish in 5 minutes. This is almost my second last slide, I think. So you can actually knock in any region in the target sequence just by linearizing it. So linear DNA is much more accessible to another linear DNA where you can. So the target DNA is also linearized knock-in DNA, right?
Yeah, we have. Yeah. Okay. Now we can see.
Yeah. Okay. I just wanted to show, acknowledge my team, as well as the activities that we do in the FLY facility at NCBS. And these are the institutes that are housed in the same campus, which we cater to. But we do services internationally. So, our funding comes from these 3 institutes, as well as Department of Biotechnology and Department of Science and Technology in India. And I am happy to take questions now.
Great. Thank you very much, Dipti. It's a really interesting and encouraging presentation for us.
Thank you.
Great. Now Dipti has shared her thought and also teach us about CRISPR-Cas9. Maybe some participants would like to ask questions. They can either ask directly by this Zoom facility to Dipti directly, or by using the WhatsApp group. Ask the question there and I can deliver the question to Dipti directly. Okay, is there anyone that want to ask question? Okay, while waiting, okay, there's one participant who wants to ask questions. She is from UK, I think. Okay, please, Alia.
Hello, Dipti. Can you hear my voice?
Yes, I can.
Thank you very much for your sharing. It's very interesting. And I have several questions. The first one is maybe pretty basic, but if you could explain more about the PAM sequence. From what I understand, that we need to target a sequence that has the PAM sequence on it. So yeah. In real setting, if we want to target this kind of sequence, whether we need to insert the PAM sequence first, or we need to find any sequence that already has the PAM sequence, because I'm thinking about if we want to introduce or delete a sequence in the biological system, we might find it hard to find this exact sequence that has the PAM sequence on it.
Okay. So Alia, PAM sequence is actually not very difficult to find, because it's only for the particular Cas9 that we are talking about, which is mostly used, which is coming from Saccharomyces pyogenes, is NGG. So it's N can be any nucleotide, and then 2 Gs should be next to each other. Now, what you would realize is that if 1/4 of your genome is consists of G, which is like if it was equal, A, T, G, and C were all equal, then finding 2 Gs are actually 1 in 16 probability. So you will actually find every 16th base to be this GG together. If it was a very homogeneous system. Now you may not find every 16th base to be GG, and it might be a little bit more more difficult, but it's not very difficult. If you just look at any sequence and just search for GG, you will see that there are so many GGs in the genome, and there is quite a lot of possibilities. Now, there are regions in the genome which are AT rich, for example, and there are regions in the genome where you may find it very difficult to find it. For this, people are actually looking for other Cas9s and also making the same Cas9 more flexible for other PAM sequences. So these are being made available. For example, CPF1 enzyme has a AT rich PAM sequence. So you can actually use that kind of an enzyme for it. So there are many there is a lot of choice of target PAMs that you can use from.
I see. Thank you very much for the answer. And can I ask another one?
Sure. You may.
Okay. So you mentioned about to validate the fly that already contains the transgenic in the method D, I think you mentioned that. Yeah. So if it's observable, so you can see the phenotype through the observation. But if it's not observable, you mentioned that you will need to sequence the what is it? The offspring of the flies. So how many trials you will need? Do you need to sequence each of the offsprings, or and you also mentioned that you can use a marker that has colors so you can see the result, but I think that maybe there's another method to validate the result in the offsprings.
Yeah. So, so when you want to remove the function of a gene completely, what I recommend to do is to remove the gene completely and not just depend on one guide RNA to make a small deletion or insertion to make it out frame shift mutation. Those are more difficult, and actually you will need to do sequencing. But what you can do is actually remove the entire gene. Let's say the gene is 2 kb. And you make a guide RNA which is at the very beginning of the gene and a guide RNA which is at the very end of the gene. So you are removing the entire 2 kb chunk. When you do that, what you can do is you can screen just by PCR. You design primers so that instead of getting 2.5 kb product, you get a 500 basis product if you're removing 2 kb. And those are the things you can actually do much more faster in a 96 well format, for example. Actually, if you are, I mean, you don't have to do that much also. I'm just, I mean, even if it was a difficult thing to do, you could. So PCR would be the fastest way to do it. Now, if you want to remove a particular domain, or if you want to change, make one domain inactive, or ATP binding site you want to remove, or something very, very specific, there is unfortunately no other way of doing it than actually sequencing it.
Okay, thank you.
Okay, thank you, Alia, for the nice questions. Maybe there is anyone who wants to ask question. Okay, Dipti, actually, we have one question here in WhatsApp group. I will deliver the questions. I mean, okay, there are two questions here. First, thank you for your interesting presentation. A simple but truly explanation about CRISPR as molecular engineering tool. The first question is, in CRISPR, as you explained before, the role of guide RNA is very important, and it's not easy to earn a good one, but for our purpose. I may want to ask about what you've been doing. Are there any tips that we have to concern about? For instance, we are going to generate a guide RNA for human medical purpose, such as disease diagnostic or therapy. Also about the guide RNA, about its properties. Are there any special handlings needed once we've gained what we want? Do you get the?
Yeah. I get the question, but it's for humans, it's slightly more complicated than what it is with flies. So with humans, you will have to see, for example, this very specific. So humans have very many SNPs. You will have to sequence the. It has to be very person-specific approach, because if you were thinking of a therapeutic approach for a human, you will have to sequence this human's genome and find which mutation you want to correct. Then in that region, whatever the sequence is it does it have off targets anywhere else in the genome? Off targets is a big issue in human genome because it's a huge genome, and whenever I have tried to look at human genome for guide RNAs, there are like 35, 40 off targets. Suddenly, when I do the same experiment for flies, there is hardly any, or maybe like, you know, 1 or 2 off targets. Also, when you are talking about humans, you are not, I mean, once you have injected a guide RNA, you have injected a guide RNA. You can't, I mean, all the off targets are going to be there. With flies, you can always use the balancers, for example, to clear up other chromosomes and so on. So actually with therapeutic, it's much, much more complicated, and hence you have to be very, very, very careful about what you're doing. The point in example are these 2 Chinese babies, right? And you know, it's very complicated. I don't want to comment too much on it, actually.
Okay, great. Yeah, the issue is still hot, isn't it?
Yeah, yeah.
Yeah. The second question from the same person. So the person who asked question, his name is Rudy, and he's working in Indonesian FDA actually. So he's our alumni and alumnus, and he is working there. The second question is, this may out of your presentation yet, still about CRISPR. Related to COVID-19, latterly FDA for the first time just approved CRISPR-based test for COVID-19 detection. And it could be alternative rapid molecular test for PCR. But what I'm curious about its potential to be used as a therapy option for COVID-19, as CRISPR originally is an immune system in bacteria due to injection, as what we've been through. I mean, we've been discussed right now. So, what do you think about using CRISPR as a therapeutical purpose?
I mean, this is not a genetic disease, so I don't think I mean, it's an infection. So unless it was a genetic disease where you could actually use CRISPR to change something in your genome and then use it, I am not very sure how this can be used. Now, having said that, knowing the way this thing functions, I mean, how it infects, and you know, there are receptors that are being elucidated that COVID-19 goes and binds this receptor, and this is how it enters and all that. I mean, this is all speculation, maybe, and that's exactly what this, again, going back to the Chinese guy, he did that where he was looking for receptor for HIV, for example. I mean, these are all speculations, maybe, but the more importantly, what the diagnostic use of CRISPR-Cas9 is, and the potential of it is humongous, because in vitro it can actually cut the genome, cut any DNA at a very, very specific genomic or a sequence specific way. So for diagnostic, it's very useful, but I'm not very sure about therapeutic as such.
Yep, great. Thank you, Dipti. So is there anyone who wants to ask questions? So basically, maybe everyone is shy. Or maybe they already understand your nice presentation, because I think what you've been saying is really, how to say, I think we can follow the presentation nicely, as what some people already said in the WhatsApp group. So we use WhatsApp to organize this event.
Okay. Thank you.
Do you use the same WhatsApp also in India?
Yes, we use WhatsApp a lot. Yeah.
Okay. There's one person who wants to ask question. Yes, please, Irma Andriani.
Yeah. Hello, Dipti.
Hi, Irma.
Hi, nice to see you here in Indonesia. Yeah. Thank you very much, Mr. Dr. Firzan, for facilitating us here to learn about CRISPR-Cas9. Actually, I have many colleagues working on CRISPR-Cas9 before. I'm working on Medaka fish Do you know, Dipti?
Yes, yes.
Medaka fish, yeah. So we have applied CRISPR-Cas9 in Medaka. And because I am in genetic laboratorium, I'm working as a lecturer and lecturer in genetic in my department, biology department. And I'm just wondering, is there any difference, a principal difference between application, the implementation of CRISPR-Cas9 technology on many species, like I mean, maybe different between Drosophila melanogaster and the on fish, on frog. Can you share about that to us?
I think
Thank you.
I think bacteria are as far to Medaka as far to flies or human. I mean, they are like very different from all the eukaryotes. So the differences between these organisms that we are talking about will not come from from that at all, like how Cas9 functions. I think the differences will come based on how you grow them. For example, what temperatures you grow them at. So the physical parts of it, because how Cas9 will interact with guide RNA is going to be same in any system. And it is also going to be same in any system how it will go and recognize the DNA. Now the differences will come from how the DNA itself is wrapped in the nucleus in any system. Also, what is the method of delivery, temperatures at which you grow them. And so these are the system specific problems. Also how the genome itself is, how big the genome is, all those kind of things. I don't see any mechanistic Cas9 mechanism specific differences in the thing, or guide RNA sequence specific differences. I don't see there should be any. It would be more of these very system specific issues that you face even when you're making something else, like making transgenic or anything. The same differences you will see, I think, in other systems as well.
Yeah. Okay. Thank you, Dipti. And I think I hope you don't mind if I can do a correspondence to you, yeah, about this.
Sure, no problem.
Is that okay, Dr. Firsan, if I directly correspondence to Dipti, yeah?
Yes, you may. Yes.
Thank you very much, Dipti, for your answer.
Thank you.
Boleh tahu Bu dari institusi mana? Your institutions.
Biologi UNHAS Biology Department.
Okay. Hi. Oh yeah, thank you. Yeah. Okay, great. Is there anyone again who wants to ask question? I think even like basic question will be nice, like the application of this technique. If I may, maybe I will ask on behalf of students, Dipti. So basically, from your presentation, we can use CRISPR-Cas9 for knock-in or knock-out, right? So maybe not many of our students or maybe participants in here realize the potential use of this CRISPR-Cas9. So can you like explain, for example, would it be possible to insert human insulin gene, for example, into the Drosophila, or another genes into Drosophila to make humanized Drosophila, for example? Would it be possible?
Yeah, it is possible. In fact, it is already being done in Hugo Wellen's lab. He has a large collection of these human disease genes that are already inserted in flies. These were not done by CRISPR-Cas9. These are transgenics. So what can now be done with CRISPR-Cas9 is actually to replace. So replace fly insulin receptor gene with human insulin receptor gene. That's a possibility. yeah, I mean, but in form of transgenics, this is already there's a huge collection of such genes already there. Yeah.
Great, that's nice to hear, because it will increase our, how to say, in Indonesia there's not so many people use Drosophila, and I would like to encourage people to use this kind of fascinating model to improve our understanding about disease, and this will be a nice introduction for that. So we have heaps of tools that may not heard by everyone, and CRISPR-Cas9 has been proven as one of the tools that is really good using in Drosophila, right?
Yes.
Yes. Okay. Another question probably is, when you try to create, for example, Cas9 lines or guide RNA lines, for example, how much percentage you can get the expected phenotype when you inject them?
So it's very experiment specific, and there are many reasons why this is so. One is that the guide RNA itself, the sequence of guide RNA itself decides efficiency. So as I said, that many empirical data suggests that having a GC-rich region proximal to the PAM site leads to higher efficiency. And there are several such rules. So sometimes it's not possible to have the guide RNA exactly of your choice. Sometimes you need this region of the genome to be mutated and no other region. And there the guide RNA efficiency is going to be low just because there is a lot of AT rich region or whatever. But you have no choice. You will have to use that guide RNA only. So that is one reason, that can be one thing. The other is also what we have also seen, and it has been reported, is that guide RNA that works very beautifully, let's say neurons, and it's highly efficient, is probably not as efficient in other tissues, like in flight muscles or in some other tissue like that, which is because maybe the way the genome is tightly wrapped around the histones in this particular, because in the end, physically thinking, it's actually Cas9 needs an access to the genome to be able to cut it. If there is, in heterochromatic regions, for example, which are very tightly wrapped, you will not be able to cut as efficiently. So all these things matter a lot, actually, and also very genes which are very highly essential. So, for example, genes without which the cell is not going to survive. Cell is going to be resistant to this Cas9. So the cells which are, the gene, for example, every time you cut the gene, the cell is going to die. Those are not the cells that are going to be selected in the next generation. The only cells that will be selected in the next generation, which have got the cut in such a way that the protein is still functional, but is now resistant to Cas9, CRISPR guide RNA. So all these are real issues, and that's why some of them are 100% efficient, while there are some where you screen thousands of flies and then you get one mutant maybe. So yeah, it's very variable, highly variable.
Yep, great. Yeah, there's one of my colleagues want to ask question. Maybe before she asks questions, I would like to discuss with you how long. I'm thinking maybe in the next 15 minutes for the discussion, if you are okay, or longer. I mean, which one you prefer?
15 minutes is fine.
Ah, great. Thank you. Miss Hasni, please, you may ask your questions.
Thank you very much, Dr. Firzan. Can you hear my voice?
Yes, I can.
Okay. Okay. It's actually, my question is very simple, very basic. Based on your introduction before, you say that between the distinct genetic background of Drosophila due to the ubiquitous of polymorphisms, it's very critical to sequence the intended target locus in the genetic background in which the genome engineering experiment will be performed. So the problem is, if there is a single base pair change in the target site, it's very detrimental to the success of the experiments. So my question is, how you overcome this kind of problem, or how to avoid this kind of problem?
Yes. So as you said, as you rightly said, that if there is only a single base, that, I mean, especially the 12 proximal bases next to the PAM, even if there is one single base difference, this guide RNA is not going to work at all. The rest 20 bases, 8 bases may be, you know, still okay. But these 12 are very essential. And that is the reason why I suggested that this region should be sequenced. So what you should do if you are, let's say, interested in insulin receptor in Drosophila, and you want to mutate the N terminus of the protein, or specifically, or you want to remove the entire gene, what you do is you take some 100 to 200 bases, you do a PCR of these 100 to 200 bases and give it for sequencing. Once you have the sequence, you can blast it and see if there are any polymorphisms in your particular fly. And if there is none, you can take that into consideration and make and design the guide RNA. If there are polymorphisms, you keep that into mind and make your guide RNA accordingly with the mutation that is there in your fly, because that's what you're going to use for your experiment.
Okay, thank you very much. If may, one more question, is it okay?
Sure. Sure,
yeah.
Sure.
Because I'm working with the wound healing, and then you said that before the Drosophila is very, near with the human. So is that any kind, is it possible to use this CRISPR-Cas9 with wound healing? Maybe like you said before, you can use rat genetic for the cornea, and then in human we have a cornea endothelial disease. Maybe we can use this CRISPR-9 to promote wound healing in corneal endothelial cells, maybe? Is it possible in the wound healing study?
You are talking about wound healing in flies, or you're talking about wound healing in cornea for CRISPR?
For CRISPR.
No, but which system?
I don't know. I'm just asking whether it's possible.
So yes, so wound healing is actually studied in flies quite a bit, actually. In fact, if I remember correctly, Andreas Prokop has a very nice video of wound healing studies in flies, and some basic kind of fun experiments to do in his education video, something like that. So to cut the long story short, yes, wound healing is studied in flies, and if you want to use CRISPR to mutate certain genes, you can do that in flies. So yes, both are possible. If that answers your question.
Yeah. Thank you very much for your answer.
Yeah. Okay.
Thank you, Ms. Hasni Hassan. So, Dipti, this is my colleague that doing her PhD in South Korea.
Okay.
Yeah. I'm thinking about her question. So we can apply CRISPR-Cas9 to maybe investigate the role of certain genes in wound healing or something like that, right? Yeah.
Yes.
Great. Nice. Is there anyone who wants to ask questions? So my work basically checking who wants to ask questions, something like that. Okay. Yeah. Maybe not so. Okay. While waiting, I want to ask you, would it be possible for example, staff or students from our university or our faculty come to your lab and learn directly how to do the CRISPR-Cas9 and having fun with it, like in the future?
Yeah,
When the COVID...
we do provide training. So yes, that's a possibility. Yeah.
Great.
On a
So
decided time and whatever. Yeah.
Yeah, that will be nice. Yeah, because hearing this and doing that will be different. Like, yeah.
Definitely. Yeah.
Yes. I did kind of like CRISPR-Cas9 based projects in Kanazawa University a little bit. We used the NOS-Cas9 at first, like the in your presentation you introduced Cas9's line and single guide RNA line, and then do the mating, something like that. It will be fun for students, I think. Yes. Okay. Maybe someone want to ask question? Okay. Maybe everyone is happy with current. Good. So we have no other people who wants to ask question anymore, probably. And based on this, I'm so happy that you accept our invitation, and this is our first, direct interaction.
Yes.
And yeah, would it be nice to ask the presentation to give to our students and staff? I mean,
I will send it to you. If you can send me your email, I can send it to you.
Yes, please. Yeah, I will send you the email later. Okay.
Yes. Sure.
Yeah. Okay. Great. So, do you have any comments on I mean, basically, this is the first webinar in the series, and then Professor Andreas Prokop that you mentioned will be the next presenter. So I invite him actually, and then he is eager to deliver the presentation on neurology, I think. Yeah.
Oh, nice.
So yeah. So
can I attend it too?
Yeah. I will give you the information by Twitter or Facebook. Yeah. So basically we are starting this webinar series based on COVID-19 problem. I mean, we cannot invite you coming directly to Indonesia, but maybe in the future, if you are happy, we can try to establish link between your institution and our institutions, and then our staff can come there, can visit your place, and you can visit our place as well. Yeah, that will be nice.
That would be nice. Yeah.
Sure. Yeah, great. So now it's already 1.5 hours of so it's really nice to have your presentation. And I got some testimonies here that said your explanation is really clear, and your presentation about the basic of the CRISPR, as I personally asked, is really helpful to make us understand about the CRISPR-Cas9. Thank you, Dipti. I'm really sorry if we cannot do any direct interaction at the moment.
No, thank you so much. And also, I'm really sorry for all that electricity problems. Just went for, yeah.
No worries. Okay. No worries. It will be we usually get the same problem in Indonesia, but yeah, thankfully there's no such thing at the moment. Yeah, I hope you can cope with the COVID-19 there, because it's kind of a problem here. Yeah. Thank you, Dipti. We will let you know about next webinar series again.
Sure. Thank you so much. Okay.
See you.
See you. Take care. Bye.
Thank you.